Thank you, and let me know if you have any questions.
Thank you very much, Srikant. First question is Tom.
Thank you for the lovely talk. What is the overall scale of the entropy production rates that you're getting in those cell experiments?
So here we got, in the best case scenario, it was about 1 ATP hydrol, so like up to 10-15 kBt per second when I integrate over the entire surface of the cell.
Okay, so
yeah,
a very small number, right?
It's a very small number. And I would expect like thousands.
So what do you think you're actually capturing?
So I know what I'm missing. It's just basically the one of the thing that we are missing is the time, sampling rate. Like, so here we are working with a sampling rate of 50 millisecond, and with the TUR estimated, that directly goes into the denominator. And there is a significant reduction in entropy production when you coarsen your process in time. So if I were to work, with not 50 millisecond, but 5 milliseconds, which now we have data, so we are going to start looking at that. I think estimate, of course, one has to then systematically get rid of the noise also. But I believe one of the limiting things was the time resolution we had in this data. And this was a set of available data, not originally fine-tuned for these experiments, so for these studies. So I think that was one of the limiting cases. And we only capture the cell membrane dynamics. So you don't see any of the other underlying processes in this experiment. So, I mean, you don't capture, once I don't have the access to any other degree of freedom, like motors or.
So if you had infinite time resolution,
Yeah.
what you'd capture is the minimum entropy production needed to produce these kind of dynamics.
Yeah, essentially keep the membrane, minimum entropy, or the estimate by which the membrane is not just thermally fluctuating. If it was a dead cell in an aqueous solution, my membrane would still have thermal fluctuations.
Yeah, but presumably your thing would say 0.
Yeah. So this method would tell apart like how much away from non-equilibrium is that membrane alone. Yeah.
So but is that right? Is it somehow saying this is the minimum entropy production you need to see this kind of dynamics?
I think so. If I understood your question correctly, yeah.
Okay,
that's
fine.
Yeah.
Okay. Jonathan?
Yeah, I had exactly the same question, where like I was wondering to what extent does your estimated entropy production match roughly about 150 picowatts of the HeLa cell? And of course, I would expect that it's much lower because you're only measuring the membrane. But I was wondering, right, like if you now, just a suggestion, you could just also do, say, how many proteins do I have in a square micron of my membrane given, and you could estimate roughly the ATP hydrolysis rate based on some k-cuts of these enzymes and see whether you roughly reach the same order of magnitude, right?
I think going forward, that is the path. So here we are clearly off by orders of magnitude, but one to make sense of the number, I think that is what one will have to do, like to figure out like what are at least the minimal settings that would need to an active membrane dynamics.
No, but that's exactly, it's the way to go. At one point, I love the approach to try to get a spatial resolution. And it's the right, I like looking at fluctuations, I think is the right way. And now we just have to see to get in the right order of magnitude somehow.
Yeah, exactly.
Depending on what we measure, right?
Yeah.
Patrick.
Hi, Srikant. So I was just wondering, in that heat map that you're showing the log of the entropy production. So that implies already that it's positive. Now the second law would only tell me that the overall entropy production must be positive, but the local one could in principle also be negative. Do you define it in a way that makes it positive automatically, or is there or could it in principle be negative as well?
No. Here, here I'm plotting the average entropy production over 10,000 frames in a particular time instant. So I talked about local entropy.
Over space, I guess even in space you could locally have a sink in entropy production, and that is outweighed by a much larger positive entropy production elsewhere.
It is possible, yeah. So if I now instead of if I make a movie of my cell membrane colored with entropy production, I didn't want to do that because I didn't believe we had sufficient data to claim you can do that. So if I do that, then you would also see fluctuating entropy production where there are reductions and increase over the membrane of this coarse grain, entropy production. So at this moment, I'm averaging over all those frames and giving you one. So each of this picture here has 10,000, about 10,000 frames averaged over, or 5 to 10,000, I think. In the lowest case, about 2,500 or so. So it is averaged over those many frames and it's giving you a number. But in principle, I have the data to be able to plot a fluctuating entropy production and make a movie of fluctuating entropy production. But just now, I think, yeah, we need more data to be reliable on it. Like, yeah.
But in the theoretical model, could it be that you just have one spot where all the entropy production is negative and another one where it is positive? And of course, the overall spatial average has to be positive.
Yeah, so here you're only seeing the overall average. That's why it's all positive in everywhere. But in principle,
it's not the overall spatial average, it's the overall temporal average. But I guess
yeah, I
there's
mean,
also the spatial variation, which in principle, I guess, could be negative.
yeah, it is
Basis.
possible. But in the way that is colored here, I'm taking each region and so a box of several spatial points, pixels. And that is for that region, I'm averaging, getting an entropy estimate for that entire
That is
region.
always positive just
That is
because
always
that's what the
positive.
data tells you.
Yeah, yeah. But it could be like with more resolution data, I could in principle obtain what you were suggesting. Like one would be able to make a movie of a cell membrane fluctuation.
But you talk about temporal resolution again. So if
Also
I had
spatial,
better
yeah.
spatial resolution,
Um,
I would still not see any negative spots there, I guess.
so each region I'm getting a lower bound from TUR, so that is by construction positive.
Yeah.
But in principle, yeah, I think it is possible to get also negative. Yeah, you can, in principle, it should be possible to see reductions in entropy production. Yeah, we can discuss it. I think it's an interesting direction. Yeah.
Thanks.
Yeah.
Okay, so thanks again to Srikant, and we move on to William Jacobs for the last talk of today. So,
all right, can you hear me and see my screen?
Yes, we see your screen, we can hear you.
Fantastic. All right, well, thank you so much to the organizers for the invitation to speak. This is certainly a wonderful conference. So today I'm going to be speaking about biomolecular condensates and how to model them out of equilibrium. Right, so condensates are essentially droplets of proteins, nucleic acids, and other biomolecules that can form inside of living cells. So there's quite a variety of different condensates that have been studied at this point. But in general, if you fluorescently label one protein or one nucleic acid component, you see the emergence of droplets that are spatial inhomogeneities. And these are driven by thermodynamic forces, surface tensions in particular, to form these demixed structures spontaneously. What I want to focus on today is the fact that in many cases, these systems are inherently out of equilibrium. Of course, they're forming inside of living cells, and there can be chemical reactions, in particular ATP-dependent chemical reactions, that modify the conformations or chemical details of these various biomolecules, and thus their tendency to form condensed phases. And so this example here from these condensates known as P granules involves both a phosphatase and a kinase that can phosphorylate a particular protein that modulates its ability to form these droplets. And this is actually one of the very first systems to be studied in this field about 15 years ago. But there's, as I said, a huge variety, and I think some of the really exciting examples are ones where the kinetics of condensate formation are essential to their function. So that this particular example here of lat cluster nucleation takes place within T cells, and the formation of these clusters on a two-dimensional membrane turns out to be important for antigen discrimination. So determining essentially a friend versus a foe antigen depends on the delay time to form one of these condensates. There's a nucleation process, and again, all of this is out of equilibrium because these lat proteins can also be phosphorylated by a pair of kinases and phosphatases that controls their nucleation dynamics and is thus essential to their role in this antigen discrimination. Right, so if we want to think about how to describe a droplet like this, the natural place to start is with equilibrium thermodynamics, and we should be thinking about, in the simplest case, a phase diagram for one component. I'll use rho throughout this talk to represent a concentration of a biomolecule. And standard in material science, we would think about a temperature-dependent phase diagram where we have at coexistence a binodal given by this black curve, where within that we have a coexistence region, meaning that a system at a temperature and concentration within this region spontaneously phase separates to form coexisting phases, one with a high concentration and one with a lower or dilute concentration of this particular biomolecule. And so our system should look something like this. If we're in the coexistence region, we have these droplets that form at equilibrium. And there's associated with this a surface tension that is also temperature dependent and vanishes at a critical point. Right. So what's useful about adding chemical reactions, most simply, is the fact that in a cell, you don't have a huge control over temperature, and producing proteins or nucleic acids is a relatively slow process, right? So introducing these out of equilibrium reactions gives you a new independent control parameter for moving around in this phase diagram. But more than that, you can also get truly non-equilibrium behaviors that don't have an equilibrium counterpart. For example, it's been proposed that you can have a size control of droplets at a non-equilibrium steady state. In the case of the T cell lat cluster formation, it's been proposed that these clusters act as a form of kinetic proofreading via nucleation. And so there's some very interesting things that can come out of coupling chemical reactions to phase separation. Right, so to actually model this and incorporate these non-equilibrium aspects, a common starting point is the framework of linear irreversible thermodynamics, which holds close to equilibrium. So I just want to walk you through what this looks like in many of these existing theories. So the key idea is that at small length scales, it's still possible, or at least it's assumed to be still possible, to define a free energy, right? So given some free energy density F, we should be able to compute a chemical potential. This is a purely equilibrium quantity, but then we would add in some chemical reactions that have some net reaction rate that's dependent on these chemical potentials. Say for two species A and B, we have a forward rate and a backward rate. And if we put all this together, we get what's known as a reaction-diffusion equation. So we have a conservative part here that involves the divergence of a flux with these mobility coefficients given by lambda. And here are chemical potentials. And then we also have these non-conservative contributions here from the chemical reactions that are taking place. If you take an equation of this form and go to a dilute limit at steady state, it can be rewritten into a more appealing form where you get this single parameter psi, which is the reaction-diffusion length scale. You can interpret this as how far a molecule can move on average before it undergoes a chemical reaction. So the challenge is, if you want to take this approach and apply it to a system with phase separation, then we need to be able to incorporate inhomogeneities. And what I want to focus on in this talk is really how do you describe this phase-separated interface, and can you use this equilibrium or near equilibrium picture? And so this is where stochastic thermodynamics comes in, because it allows us to develop a microscopic model to ask the question, what are appropriate interfacial boundary conditions in this case? What happens beyond the linear response regime? And in particular, what can we learn about fluctuations? Because this is incredibly important for being able to describe the kinetics of an out of equilibrium phase separation process. So the model that I'm going to discuss is arguably the simplest possible model that has the relevant ingredients here. So we have molecules in an implicit solvent with the addition of some enzymes and a constant chemical fuel source. So we're going to use B and I to represent two possible states of a molecule that are either a sticky phase-separating state B or an inert state that is otherwise energetically equivalent to the implicit solvent. They differ in their internal energy by some delta F. So this would be an equilibrium pathway for the chemical reactions. We also are going to have a non-equilibrium pathway that is driven by an additional chemical potential difference, which could be ascribed to a chemical fuel and waste, right? So the system is imagined here to be chemostated. There's a constant concentration of fuel versus waste, and that gives us a non-equilibrium steady state. Okay, so within the language of stochastic thermodynamics, we are assuming here that the solvent and the chemical fuel and waste are all at equilibrium with some inverse temperature beta. And local detail balance then gives us constraints that have to be obeyed on both of these reaction pathways, so that the passive 1P and the driven 1D both have to obey these relationships between forward and backward rates. One depends only on the internal energy difference and what else is interacting with these molecules, represented by this potential energy U. But the driven pathway also has this contribution from the chemical drive. All right, so we are implementing this, again, for the sake of a minimal model in a lattice context. So we have diffusion. So molecules can hop to nearest neighbor lattice sites. And they can also undergo chemical reactions. And the orange molecules here, which are the phase separating B state, participate in nearest neighbor interactions with an interaction energy epsilon. So since we have access in a simulation of this form to all of the microscopic details, it's straightforward to compute the time-averaged steady state entropy production rate. And you immediately see that it's spatially dependent. So this should make sense because the local environments in the two different phases are quite different. In particular, you'll see that the entropy production rate is lower in the condensed phase, right? So this is our droplet here. And that's because these B state molecules have attractive interactions with their nearest neighbors, which suppresses reactions and thus reduces the overall entropy production rate here relative to the dilute phase. So if we want to ask a question at a more coarse-grained level about mesoscopic properties, then we can look at this exact same system and look as a function of the distance from the center of the droplet. So our average interfacial position is at z equals 0 here. And we're interested in a net diffusive flux, right? So we want to think about, for the system just shown, assuming that the the chemical drive is the same everywhere, which gives us the same reaction rate constants everywhere, but clearly a non-uniform entropy production, how are the molecules actually moving on average? And it turns out for this particular system that there's no net flux, which may not be surprising to this audience because we're thinking about a coarse-grained property of the system. And in this particular case, we're essentially losing the signature of entropy production when we coarse grain in this way for this particular set of conditions. Right? So to actually be able to study a more interesting system where we have mesoscopic fluxes that are present in our phase-separated system, we can consider making these chemical reactions spatially inhomogeneous. Right? There are many different ways this could happen. One possibility that appeals back to these motivating examples at the beginning would be introducing an enzyme that preferentially partitions into one phase or the other. So let's say that there's an enzyme that's essential for the driven pathway and that it partitions into a condensed phase. So it looks something like this picture here with my cyan enzymes in the condensed phase. That would make it so that my reaction rate constants would no longer be the same for all possible local environments. In particular, they'd be higher in the condensed phase. And so, again, obeying local detail balance gives you this relationship between the forward and backward rates and the driven and passive pathways. Right, so this is still going to give us entropy production everywhere. We want to ask, where do we actually see mesoscopic fluxes? Right, so again, this is our picture. We have an interface at z equals 0 between a condensed and a dilute phase. The interface itself is fluctuating. So if you look at a time average, you see this sort of smeared out profile. But now, if you track the two different species of molecules, you do see net diffusive fluxes, right? So on average, the B state molecules that want to phase separate are flowing into the condensed phase. The I state molecules that are inert are flowing out. And the reactive flux is also similarly concentrated near this interface, right? So the key point here is that if you go far from the interface, you can always define an effective equilibrium, right? There's still entropy production, but overall, you don't see any net fluxes. But right in the vicinity of this interface, you can see this coarse-grained signature of non-equilibrium behavior. You can further break down where these contributions to the various fluxes are coming from. So some of this is happening in the dilute phase, and that's shown here by these dashed lines. This is well described by this reaction-diffusion equation in the dilute phase. But the thin solid line here shows you the contribution from reactions that are taking place actually on the interface itself, right? So really tracking what is happening on this fluctuating interface is important for describing the system here. So all of that, again, is at this coarse-grained level. If you look at a more microscopic picture, you can ask questions about how the interface itself is changing by being driven out of equilibrium. One thing to look at is capillary fluctuations. So if you look at a slice of our system, you'll see that we have this fluctuating interface between the condensed and dilute phase. These waves are referred to as capillary fluctuations. And for an equilibrium system, as shown by these dashed lines here, you get this 1 over q squared scaling, which comes from the equipartition theorem. When the system is driven out of equilibrium, this fluctuation spectrum can change. It's possible to see that the amplitude overall can be increased or decreased depending on the setup of the system. It's also possible to break this equipartition theorem relationship. That's one thing that we can look at. We can also look at the actual statistics of the molecules that are sitting right on the interface itself. Right, so if you take the Boltzmann relationship and invert it, you can define an effective internal energy difference between the B and I states. Right, so this is dependent on the number of B state molecules around them because that affects their potential energy. And at equilibrium, this is just equal to this delta F that is an input to the simulation. So this has to be the case according to the Boltzmann distribution. But for our systems that are driven out of equilibrium, we can actually see systems where this effective internal energy difference is now environment dependent, which means, in other words, that we have non-Boltzmann statistics, right? There's no single effective equilibrium model that can possibly describe everything that's happening on this interface, right? So even at this short scale, this is not a short length scale. We have a system that is clearly out of equilibrium. Right, so all of this gives us an approach to try to predict these effects from a theoretical point of view. So I'll just quickly walk you through what we've done here and try to answer the initial question that I posed. So in the dilute phase, we can use this reaction-diffusion equation. But our new contribution is to think about a microscopic model of the interface. So we're essentially writing down a master equation that captures the microscopic rates that we have in our simulation. So we say that the interface has to be at steady state. So we have this J contribution here, which has to do with diffusive fluxes, and this S contribution that has to do with reactive fluxes. And what we want to do is match these two descriptions to each other, right? So we have a mesoscopic description in the dilute phase, a microscopic description of the interface. The fluxes have to match, right, for mass conservation, right? So we have our ensemble, steady state ensemble averaged diffusive fluxes have to match what's happening in our mesoscopic picture in the dilute phase right at the interface. And then we also have to say that there's no net growth of the condensed phase, right? So the interface, in other words, should not be moving on average in the course of the simulation. And based on some prior work, we've shown that this expression here, which you can argue on symmetry grounds, provides a reasonable approximation for this non-equilibrium supersaturation. And we relate that to this, this term that has to do with the curvature of the interface. Okay, so putting all this together, this allows us to predict non-equilibrium phase diagrams. So I'm showing you here for the case of a macroscopically flat interface. So in other words, a gigantic droplet where the interface looks locally extremely flat. If you always have the same concentration of molecules in the far field, then you have to tune this internal energy that distinguishes the B and I states in order to remain at coexistence. And that's shown by these points from simulations, these curves from the theory under two different conditions, as you change the mobility of the molecules, right? So molecules move much more quickly related to, by comparison to reactions as you move to the right on this plot. And this also allows us to predict what these fluxes are near the interface. And it's useful to contrast this with what you would get if you assumed that the interface itself was always at equilibrium, right? So it's possible to write down an alternative theory where you have reaction diffusion in the bulk phases, but you assume that the interface itself is at equilibrium, and that's shown by these dashed lines here. And this really only works in this linear response regime, which is when you take this mobility to be very large. Right? But in a more general case, this microscopic description of the interface that we're introducing here turns out to be essential for accurately getting these properties. And really the overall message that we learn from these microscopic studies is that non-Boltzmann fluctuations at the interface are essential for connecting, for being able to observe mesoscopic fluxes in the bulk phases, and also lead to these non-equilibrium interfacial properties, including these capillary wave amplitudes that differ from equilibrium. Right. So the key point here is that at all scales of these systems, you are out of equilibrium. If you have non-equilibrium interfacial properties, then you have steady state fluxes and vice versa. And the last thing that I want to point out is that it's really essential to describe these non-equilibrium interfacial properties if you wish to describe the properties of finite size droplets. So the well-known Young-Laplace equation, or Gibbs-Thompson condition for curved droplets, says that when you have a finite size droplet with radius r, the coexistence condition, so the concentration of molecules outside of the droplet, differs from what you have in this thermodynamic limit due to a pressure difference across the interface. And that's described by this Young-Laplace equation, which has an interfacial tension over R. But because the capillary fluctuations are changing out of equilibrium, this interfacial tension also changes. And so when you do these simulations for different non-zero values of this chemical drive, then you get a different interfacial tension that's required to be able to describe what's happening in the simulations. And these interfacial tensions are related to these capillary fluctuations that we see from those spectral measurements, right? So it's essential to calculate these interfacial properties in order to predict what happens with finite size droplets. And there's no place that's more important than when studying nucleation, because nucleation of droplets is inherently a fluctuation-driven process. And so the ability to be able to tune the interfacial tension gives us an entirely different way of controlling these nucleation rates. So what's shown here in this case is a comparison of nucleation simulations with all done at the exact same supersaturation. So the effective thermodynamic driving force for phase separation is the same in all cases, but by tuning this interfacial tension, it's possible to dramatically, by many orders of magnitude, change nucleation rate. All right, so to sum up, fluctuations matter. I've shown you that stochastic thermodynamics allows us to model these types of systems far from equilibrium. When there are mesoscopic fluxes in these systems, they're concentrated near interfaces. But it's really these non-Boltzmann fluctuations at the interfaces that are underlying all of this interfacial properties that are changing and fluxes at larger scales, and they're all coupled together. And so I think the most important thing for the biology here is understanding how these driven chemical reactions allow us to consider altered nucleation rates in ways that are just simply